Hello and welcome. Today let's talk about the lymphatic system and we'll look at immunology here as well. Now when we go through and we talk about the lymphatic system, I would like you to know that the lymphatic system consists of lymphatic vessels, it consists of lymphoid organs, and it consists of lymph. This system is going to be closely associated with our cardiovascular system, and we talked a little bit about that in that chapter when we discussed cardiovascular system. This system has three main functions we can check out here. Number one is to return excess tissue fluid, or we call lymph, to the bloodstream. Number two, we can see here, is absorbing fats from the digestive tract. So this lymphatic system is responsible for absorbing fats from our digestive tract and then transporting them to the bloodstream. So when we eat fats, they get absorbed in our intestine by the lymphatic system, which will then transport them to the bloodstream. And then the third function we can check out here is to defend the body against disease. To defend the body against disease. This last function is going to be mainly carried out by white blood cells that are going to be found in the lymphatic vessels, in the lymphoid organs, and in the blood. So let's look at then our lymphatic vessels first. Now, when we talk about our lymphatic vessels, I would like you to know these lymphatic vessels are going to form a one-way system that will begin with the lymphatic capillaries. They will form a one-way system that begins with the lymphatic capillaries. So let's start with the lymphatic capillaries then. When we talk lymphatic capillaries, I would like you to know the lymphatic capillaries are tiny, close-ended vessels whose walls are going to consist of simple squamous epithelium, and they absorb excess tissue fluid. We can also see these lymphatic capillaries are going to be found in most areas of the body. They're going to merge into larger vessels. These larger vessels then carry lymph into the thoracic and lymphatic ducts. Let's talk more about lymph then. So these lymphatic capillaries, they absorb excess tissue fluid, which we said is referred to as lymph. So these vessels we can see are going to return lymph back to the bloodstream. Now, when we talk about this tissue fluid is mostly water, but it also contains solutes such as nutrients we can see, electrolytes, and oxygen, which is derived from the plasma. There are also cellular products here, hormones we can see, enzymes, and waste products that are going to be secreted by cells. Again, this fluid is going to be called lymph. We said the lymphatic capillaries are going to unite to form lymphatic vessels. These lymphatic vessels are going to enter one of two ducts. They're going to enter either the thoracic duct or they will enter the right lymphatic duct. So these vessels are going to merge before entering one of the two ducts, the thoracic duct or the right lymphatic duct. Now the larger thoracic duct is going to return lymph that's going to be collected from the body in areas below the thorax, the left arm, and the left side of the head and neck. And it'll return this lymph to the left subclavian vein. It'll return this lymph to the left subclavian vein. Here we can see it'll return lymph collected from the body that's below the thorax, left arm, the left side of the head, and the neck, and it's going to return it to the left subclavian vein. Now, when we talk about the right lymphatic duct is going to return lymph from the right arm, the right side of the head and the neck, and it'll return this lymph into the right subclavian vein, Lymph is going to make its way through the lymph nodes on its way back to the cardiovascular system. Here we can see we've got lymph nodes that are placed in certain areas, and this lymph, as it's passing through the vessels, it'll pass through the lymph nodes, and then it'll continue through the vessels onto the right and left subclavian veins. So you can see the right lymphatic duct emptying into the right subclavian vein, and then here we've got the thoracic duct emptying into the left subclavian vein. So thoracic duct, and we saw the right lymphatic duct. Next, let's talk about the structure. When we talk about the structure of the lymphatic vessels, I'd like you to know that the structure of the lymphatic vessels is similar to the structure of veins. It is similar to the structure of veins. Lymph is going to travel through the vessels like blood travels through the veins. We also have valves here that will help ensure lymph travels in one direction, that's towards the cardiovascular system, and then also the placement. Vessels will be found placed between muscles. So let's check that out. One-way valves, we can see, prevent the lymph from flowing backwards. And then the movement of lymph is going to be dependent on skeletal muscle contractions. Next one is the term edema. I'd like you to know edema is going to be swelling that's caused by the accumulation of tissue fluid. It's caused by the accumulation of tissue fluid. It occurs if too much tissue fluid is made or if not enough is drained away. This can lead to tissue damage and even tissue death. So it's localized swelling that's caused by the accumulation of fluids in the tissues. Too much fluid being made or not enough being drained away. So here we can check out then our lymphatic capillaries in greater detail. You can see they're blind-ended. Okay, this is where they begin. And then here lymph you can see is going to flow in one direction. And then here, once these lymphatic capillaries start to merge, they give rise to bigger vessels. And eventually when we get to our lymphatic vessels, here you can appreciate their structure and you can see they've got valves inside of them. That's going to ensure we have unidirectional flow. And you can see that flow is going to be making its way through then the lymph node. So the lymph node will check out its job and what it does to this lymph, and it'll continue on its way towards the cardiovascular system. Next then, let's check out our lymphoid organs then in greater detail. And let's talk about our lymphoid organs. Now, when we talk about our lymphoid organs, they contain a large quantity of lymphocytes. Lymphocytes are going to be the type of white blood cell that's responsible for adaptive immunity. Next here then we can see the classification of our lymphoid organs. When we talk about our lymphoid organs, we can see our lymphoid organs are going to be classified into two different classifications. They can be classified as either being primary lymphoid organs, or our lymphoid organs can be classified as being secondary lymphoid organs. Let's talk about our primary lymphoid organs first. Now, when we talk about our primary lymphoid organs, this will include organs such as our red bone marrow and our thymus. Red bone marrow and our thymus. These will be sites where our lymphocytes are going to develop and mature. Where they will develop and mature. And then when we talk about our secondary lymphoid organs, our secondary lymphoid organs will include lymph nodes and our spleen. These are going to be areas where some lymphocytes become activated. These are sites where our lymphocytes become activated. Let's talk then in greater detail about our primary lymphoid organs. So again, red bone marrow, we can see inside of the bone, and the thymus is what we're talking about when we're talking primary lymphoid organs. And then secondary, we said, we've got our lymph nodes, and we've got our spleen, which we can find here in the abdominal cavity. So let's go through and let's talk about our primary lymphoid organs then in greater detail. And here then we'll discuss first red bone marrow. Now when we talk red bone marrow, I'd like you to know red bone marrow is going to contain connective tissue fibers and stem cells that are going to be forever capable of dividing and producing blood cells. In children, most bones will contain red bone marrow, But in adults, we'll see it's going to be present only in the bones of the skull, the sternum, our ribs, our clavicles, the pelvis, and the vertebral column. These cells develop in the bone marrow, and then T lymphocytes, our T cells, they migrate to the thymus where they become mature. So we kind of describe the thymus as high school for the T lymphocytes. Kind of one way to remember that, easy way, you can think of it as we go to high school to mature. I didn't definitely mature in high school until after high school is when life really woke me up. But you can think of it as T cells are going to high school, the thymus, because that's where they go to mature. Now, when we talk about our T cells, we can see our T cells, they migrate from the bone marrow and they reach the thymus. Now, when we talk our B cells, the second major cell type here, they're going to remain and mature in the bone marrow. Next, then we have our thymus. So the other organ is the thymus. The thymus is going to be found located in the thoracic cavity between the trachea and the sternum above the heart. Above the heart. The thymus grows around puberty and then it shrinks as we get older. It's going to contain T cells and supporting cells. So here we can see it's a soft, bilobed organ. It's going to be the site of T cell maturation. Again, this is where the T cells come to mature. And we can see here, this is also going to be the site where T cells that are capable of reacting to the body's own cells undergo apoptosis. They must distinguish self from non-self so they don't attack our cells. Next, then, let's check out our secondary lymphoid organs. Now, when we talk about our lymphocytes, I'd like you to know our lymphocytes, they're going to migrate from the blood into our secondary lymphoid organs. Our lymphocytes, they may encounter foreign molecules or cells, where then they will proliferate and become activated. When we have activated lymphocytes, they re-enter the blood, where then they're able to search for sites of infection or inflammation. So first, let's check out then our spleen. When we talk about our spleen is going to be located in the upper left side of the abdominal cavity, behind the stomach. The spleen is going to be divided up into red pulp and into white pulp, mostly red pulp. And red pulp is going to be the region where macrophages remove old and defective blood cells. The spleen, we said, also contains white pulp. This is going to be lymphatic tissue where lymphocytes can react to foreign invaders. The spleen sometimes may need to be removed surgically due to trauma or disease. Although other organs can mostly replace the spleen's function, a person with no spleen is going to be more susceptible to certain types of infections. The spleen also removes older blood cells and platelets, and the number of both types of cells can increase too. And then next we have our lymph nodes. We talk about our lymph nodes are going to be small ovoid structures that are going to be found along the lymphatic vessels. They're going to be found along the lymphatic vessels, and they are structures through which lymph must pass. Each lymph node is going to be packed with B and T cells. Macrophages, they also occupy the nodes and engulf pathogens. When we talk pathogens are going to be disease-causing agents such as viruses and bacteria. Our lymph nodes are going to be named for their location on the body. For example, when we talk inguinal lymph nodes, they're the lymph nodes that are going to be found in the groin area. We talk axillary lymph nodes, they're going to be lymph nodes found in the armpit region. Our lymph nodes are going to be felt for swelling. They're felt for swelling by your physician for any signs of infection. I'd like you to also know cancer cells. Cancer cells have the ability to use the lymphatic system to metastasize and move far from their origin. All right, next then let's talk about innate immunity. Now, when we talk innate immunity, I'd like you to know the lymphatic system is going to work with the immune system to protect our body. Now, the term immunity, it refers to a condition where the body is going to be protected from various threats like pathogens, toxins, and even cancer cells. There are two main types of immunity eventually we're going to know about. First is innate, and then second we're going to see is adaptive immunity. Now, innate immunity is going to be fully functional without previous exposure to these substances such as pathogens, toxins, cancer cells, or any other invaders. Now, when we talk adaptive immunity, we'll see adaptive immunity is going to be initiated and amplified after specific recognition of these substances. Mechanisms of innate immunity are divided, we'll see, into four groups. They'll be divided into four groups. We have physical and chemical barriers, number one. Inflammation, number two. Number three are going to be our phagocytes and natural killer cells. And then number four are going to be our protective proteins. So let's go through and let's check these out in greater detail. When we talk our physical and chemical barriers, we have skin and we have our mucous membranes. The skin is going to be found protecting us on the outside. Our mucous membranes, they're going to be found lining our respiratory tract, our digestive tract, and our urinary tracts. And what the mucous membrane is going to do is it's going to act as a mechanical barrier. It will act as a mechanical barrier to the entry of pathogens. When we talk about the secretions, we'll see the secretions of oil glands. They're going to contain chemicals that have the ability to kill bacteria. The stomach has an acid pH. This acid pH is able to also kill many bacteria. And this will also include the various bacteria that are going to normally reside in the intestine and other areas such as the vagina. Number two, then we have inflammation or the inflammatory response. Now, what happens here is inflammation is going to wall off infections. An inflamed area is going to have four cardinal signs of inflammation. And these signs will include redness, heat, swelling, and pain. They're going to be caused by changes in the capillaries in the damaged area. Next, I want to talk to you guys about inflammation cells. Now, when we talk about inflammatory cells, we're going to have one type first. They're going to be called mast cells. Mast cells, they're going to be responsible for releasing histamine. Histamine is going to be a chemical that's going to promote inflammation. And there's also other cells in inflammation, and they're going to include our macrophages and our dendritic cells, which are phagocytic cells. We can check all these cells out and how they function then here in this image. Here we can see injured tissue cells and mast cells. They're going to release histamine. Histamine is going to cause our capillaries to dilate and it increases blood flow. And then our resident macrophages and dendritic cells, they're going to phagocytize pathogens and they release cytokines, which is going to stimulate the inflammatory reaction. And here we can see neutrophils and monocytes, which then become macrophages, squeeze through the capillary wall and they phagocytize pathogens. And here in step 4, we can see if blood vessels are damaged, blood clotting comes into play. Blood clotting is going to help wall off the capillaries and prevent blood loss. All right, next then let's check out our phagocytes and natural killer cells. So here we can see our phagocytes and natural killer cells. Our phagocytes are going to be cells that migrate through the walls of dilated capillaries. And they're going to engulf pathogens by forming an endocytic vesicle. And then we can see those vesicles will fuse with lysozymes, which are digestive enzymes. Now, when we talk phagocytes, this will include neutrophils and monocytes. Neutrophils are usually the first white blood cells to enter an inflamed area, and they accumulate to form pus. The second type are our monocytes. We'll see as the inflammatory response continues, monocytes migrate from the blood. Then, when in the tissue, these cells are going to become macrophages. They'll become macrophages. The natural killer cells, they kill, or you can say they target virus-infected cells and cancer cells. They are large, granular, lymphocyte-like cells. And again, they kill some virus-infected and cancer cells by cell-to-cell contact. They seek out and kill cells that are lacking a particular type of self molecule on their surface. And they do not recognize specific antigens. The fourth mechanism then we can check out are going to be our protective proteins. Our protective proteins form the complement system. This is a system of blood plasma proteins that can amplify inflammation. It can attract phagocytes, and it can bind to the surface of pathogens to ensure they are phagocytized, and other proteins can join to form a membrane attack complex. This membrane attack complex we can see here is going to form holes. It's going to form holes on the surface of bacteria and some viruses, allowing fluids and salts then to make their way into the cell, causing the cell then to be destroyed. I used to refer to this as the Big Mac. The Big Mac, the membrane attack complex. So just, one way to remember. Again, here we can see then the complement system. It will complement certain immune responses, and also it forms membrane attack complexes. And next here we have interferons. I'd like you to know about interferons are going to be proteins that are going to be produced by infected cells, and they're going to warn non-infected cells. Next on, let's move to adaptive immunity. Now, when we talk about adaptive immunity, I'd like you to know adaptive immunity is going to come into play when our innate defenses have failed to prevent an infection. So if our innate defenses have failed, then our adaptive immunity will come into play. The adaptive immune system eliminates antigens from the body. An antigen we'll see is going to be any molecule or a marker from a bacteria or other foreign substances that stimulates an adaptive response. When we talk adaptive immunity usually takes 5 to 7 days to become fully activated, and once activated, you can see it can last for years. The adaptive immune system depends primarily on B cells and T cells. Both of these cells are capable of recognizing antigens because they have specific antigen receptors. B cells we'll see become plasma cells, which then secrete antibodies. Defense by B cells is also going to be called antibody-mediated immunity. We'll see some B cells, they become memory B cells, and they're going to function then in long-term memory. Now here we can see our clonal selection theory states that only a few cells within a population of B and T cells have antigen receptors for any specific antigen. Selection is going to occur when an antigen binds to the receptors of a particular lymphocyte. It causes the lymphocyte to divide repeatedly, forming clonal cells. Only cells that have a receptor specific to an antigen are going to divide in the process. Here we can see our B cells are going to be activated in the lymph nodes or our spleen when the B cell receptors bind specific antigens. Cytokines are going to be secreted from our helper T cells, and these cytokines, they're going to stimulate B cells to divide. And we'll look at all of that. Defense by B cells, we said, is going to be called antibody-mediated immunity. Also called humoral immunity because the antibodies are going to be found in the blood and lymph. Our activated B cell clones, most we said are going to become plasma cells, and they're going to be involved in the mass production of antibodies. Others are going to become memory cells, and they're involved in long-term immunity. So if an antigen returns, memory B cells divide and they quickly give rise to more plasma cells. So here, let's check all that out. Here we can see we've got antigens, we've got our B cells, and here we've got our B cell receptors. You can see they're receptors of various types. So depending on the type of antigen that you have, it's only going to be able to bind to specific cells. Now here we can see we've got now activation occur. Here we've got clonal expansion now that's going to take place. Here you've got the antigen binding to the B cell receptor, and only the cell the antigen binds to is the cell that's going to be undergoing expansion. So here you can see cytokines from our T cells are going to come in, and we said they're going to be involved with clonal expansion. Here we can actually see clonal expansion occur now. Here we've got the antigen binding to the specific receptor, and we're going to see only that specific cell is going to undergo clonal expansion. And also you can note here, each of these cells have just specific receptors. So this cell undergoes activation. Here we can appreciate our cytokines from our T cells, and they're going to stimulate our B cells to divide. And here we can see then we've got clonal expansion occurring, and we've got memory B cells formed. So again, same process. So we've got memory B cells formed, or we said we can have plasma cells formed, and they're going to then produce antibodies. Now we can see here we've got the same thing occurring. An antigen is going to bind to the antigen receptor of only one type of B cell or T cell, and then this B cell or T cell is the only one that's going to divide, forming clones of itself, you can say. Only the B cell with the BCR shape that fits the antigen undergoes clonal expansion. Most clones are going to become plasma cells, okay? But some become memory cells. So here we can see we've got the plasma cells formed, but some also become memory cells. Same thing we've got going on here. Next, then let's talk about our antibodies. Now we talk about antibodies. Antibodies are also called immunoglobulins, abbreviated IG, immunoglobulins. Immunoglobulins are going to be Y-shaped molecules. They are Y-shaped molecules with two arms. Each arm is going to have a heavy, or we can call it a long, polypeptide chain, and a light, or we can say a short polypeptide chain, and an antigen binding site, and an antigen binding site. When the antibody binds with the antigen, it can be engulfed by a neutrophil or a macrophage. Antibodies may also neutralize viruses or toxins. And each chain has two regions, a constant region and a variable region. The constant region is going to be constant within the same class of antibodies, and the variable region is going to vary between antibodies. This is the region that binds to the antigen. So let's actually look at that structure. And here we can appreciate the structure of our antibody. So we've got two chains. We've got a light chain, and then next to that we can appreciate the heavy chains. Here we've got then the heavy chain. Here we can appreciate how the heavy chains are connected. And then we can appreciate how each chain has a constant and a variable region to it. A constant and a variable region. And then right inside of here, we've got the antigen binding sites. Our antigen binding sites. And here we can appreciate a computer model of an antibody as well. Now, when the antibody binds with the antigen, it can be engulfed by a neutrophil or by a macrophage. Antibodies may also neutralize viruses or toxins by preventing their binding to our cells. There are 5 major classes of antibodies in humans that I would like you to know about. Here you can see we have IgG first. IgG is the main antibody type in our circulation. It binds to pathogens and it activates complement and it enhances phagocytosis. Next we talk IgM. IgM is an antibody type that's found in our circulation as well, and this is the largest antibody. It also activates complement and it clumps our cells. It causes our cells to clump. IgA is the main antibody type in our secretions, such as saliva and milk. It's going to prevent pathogens from attaching to our epithelial cells in our digestive and respiratory tracts. Fourth, we have IgD. IgD is an antibody type that's found on the surface of our immature B cells. IgD's presence signifies readiness of our B cells to respond to antigens. And then fifth, we have IgE. IgE is the antibody type found as antigen receptors on eosinophils in blood and on mast cells in tissues. And IgE is responsible for immediate allergic response and protection against certain parasitic worms. We have 2 major types of T cells we're going to talk about. T cells leaving the thymus have a unique T cell receptor. They're unable to recognize an antigen without help. So they have to be presented to the T cell receptors by a major histocompatibility complex protein that's going to be found on the surface of another cell. We have 2 types of T cells. First, we have helper T cells, also known as TH cells, and we have cytotoxic T cells or TC cells. Now, when we talk about how these cells function, what we can do is we can move right over to basically this slide here, this slide here. You've got everything written down in here. I'll describe all this here. And then here we can see all the events. Now here I'd like you to know when these cells are going to respond, this is going to be known as cell-mediated immunity. Now when our B cells and our T cells get involved, this will be known as cell-mediated immunity. They are going to bind to antigens, but they, we said, have to be presented by MHC proteins that are going to be found on the surface of other cells. Now, T helper cells, they only recognize antigen from APC or antigen presenting cells with MHC class II molecules on their surface. Then second, our cytotoxic T cells, they only recognize antigens presented by various cell types with MHC class I molecules on their surface. Our antigen-presenting cells will include dendritic cells and our macrophages. They secrete cytokines which activate cytotoxic T cells and B cells, plus they're going to have other functions. Here we can see more about our cytotoxic T cells, and I want you to read more about our helper T cells also. Now, our cytotoxic T cells are going to provide immunity against virus-infected cells and cancer cells. Our cytotoxic T cells have storage vacuoles. They also have perforins and granzymes. Perforins will form pores in abnormal cell membranes, and granzymes are going to induce apoptosis. Some T cells may become memory T cells, and they may live many years, and they can quickly jumpstart our immune response to previous antigens. Next, then, let's talk about active versus passive immunity. Now, when we talk active versus passive immunity, immunity occurs when the body is protected from various threats, we said. Adaptive immunity can be induced actively or passively. In active immunity, the individual alone produces an immune response against an antigen. In passive immunity, the individual receives antibodies or cells from another individual. Active immunity usually develops normally after a person is infected with a pathogen. Active immunity can also be induced artificially when a person is well, so that future infections will not take place. To prevent infections, people can be immunized, or we can say vaccinated, against these infections. Immunization is going to involve the use of vaccines, which are going to be preparations that contain an antigen to which the immune system responds. Vaccines are going to be the pathogens themselves which have either been killed, weakened, or they are no longer virulent, or we can say they are no longer able to cause disease. Here then we can check out active immunity then in greater detail. So we said here it develops naturally after a person is infected with an antigen, or it can be induced artificially through immunizations, and it depends on the presence of memory B cells and memory T cells. So we talked vaccines, we said it's a substance that's going to contain an antigen to which the immune system responds. And they're genetically engineered microbes that mass produce antigens. So here we can see we've got a vaccine or immunization schedule telling us which age we should be getting which immunizations. It shows us the vaccines we should be taking and also the various times each vaccine should be given. Because we can see the first exposure is going to rise the titer levels slowly, and the second exposure is going to rise the titer levels rapidly and to a higher level. And then that as well we can see can eventually start to decline, requiring us to get then even more vaccines for further protection. So here we can appreciate the same thing. Next, let's talk passive immunity. Now, passive immunity is going to occur when an individual receives another individual's antibodies or immune cells. Newborn infants, they're going to be immune to some diseases because antibodies have crossed the placenta from the mother's blood, such as IgG. Antibodies IgG and IgA, they're secreted into breast milk. That's why breast milk is important for babies. Antibodies can also be injected by a physician as well. So here we can see passive immunity is going to be received from another person's antibodies or immune cells. And here we can appreciate some common natural processes where we saw some antibodies are able to cross the placenta, but they're only going to last for a few months. And then also we said some are going to be found in breast milk, which is going to help prolong immunity in the newborn. And last, here we can see it's temporary because there are no memory cells. It's only going to be used to prevent illness in an exposed individual. Here we can see antibody IgG and how it's able to cross the placenta. And then antibodies IgG and IgA are secreted into breast milk. And then last, here we can see antibodies being injected by a physician into a patient. All right.